Welcome everyone, and thank you for joining to today's webinar, Population-Based Genomic Newborn Screening, Results of BabyDetect Study. My name is Gemma Garcia, and I am the EMEA Regional Marketing Lead at Bioscience, and I will be the host for you today. Now, I am delighted to introduce you our speaker for today, Professor François Boemer. Professor Boemer is head of the Newborn Screening Center and the Biochemical Genetics Laboratory at the University Hospital of Liège in Belgium, and he is also founding member of the Metabolics.be, member of the International Society for Neonatal Screening, and of the Royal Belgian Society of Laboratory Medicine. He is author of more than 80 papers and is highly recognized for his work in developing innovative screening methods for diseases like spinal muscular atrophy, and sickle cell disease, and has been actively involved in advancing sequencing approaches for neonatal screening. Today, François will walk us through what is the BabyDetect project, a pilot study in Belgium that aimed to incorporate advanced genetic testing into newborn screening. How the study uses deep sequencing to screen for 165 treatable pediatric disorders across 405 genes with data from over 6,000 families, gathering significant data on pre-symptomatic genetic variants. Finally, he will showcase the key findings from the project and highlights of the importance of combining genetic and biochemical methods for effective newborn screening. François, thank you so much for being with us today. Now I want to give you the stage.
Hello everyone, and welcome to this webinar on the results of the BabyDetect study. BabyDetect is a clinical trial designed to implement genomic newborn screening. So let us begin by defining what are rare diseases. A rare disease affects fewer than 1 to 2,000 individuals. More than 6,000 rare diseases have been described so far, and it is estimated that around 300 million people worldwide are affected by rare disease. Most rare diseases are genetic, and about 70% manifest during childhood. They represent a considerable medical, economic, and psychosocial burden for patients and families. Many patients must consult multiple specialists before a diagnosis is done, a diagnostic odyssey which typically lasts several years. And currently, about 6% of rare diseases are treatable, but for some of them, it's not longer a fatality thanks to newborn screening. What is newborn screening? It is a public health initiative that aims to identify and treat serious congenital conditions from birth with the goal of preventing the onset of usually irreversible symptoms. To put it somewhat simplistically, but in a way that helps frame the idea, newborn screening is to rare disease what vaccination could be to infectious disease. To be included in a newborn screening program, a condition must meet several criteria, including an effective treatment must exist, a reliable and affordable diagnostic test must be available, and the cost-benefit ratio must be favorable. How does newborn screening work? A few drops of blood are collected from every newborn onto a filter paper card. Once dried, the card is sent to a centralized laboratory where analyses are performed to detect the conditions covered by the screening program. If the result is positive, the newborn is recorded for confirmatory testing and, if necessary, initiation of treatment. The use of dried blood spots on filter paper has been the cornerstone of successful newborn screening programs worldwide for over 60 years. This support is standardized, logistically convenient, and ensures good sample stability. Today, most newborn screening programs worldwide focus on no more than 50 conditions. The screening relies on biochemical markers quantified using either high-throughput analytical techniques such as mass spectrometry or immunoassays. As a result, nearly all infants born in industrialized countries and in many low and middle-income countries benefit from some form of newborn screening, through the number of conditions screened varies greatly depending on national policies. In the European Union, for example, over 3.5 million newborns are screened each year, but programs differ widely. In Italy, for example, up to 50 conditions are included in the national programs, while in other countries only one may be screened. Now, why does newborn screening matter? Here is an example of three little girls affected by a glutaric aciduria type 1, an inborn error of metabolism. Two girls on the right and the left benefited from newborn screening and received preventive treatment. The girl in the center was not screened and could not be treated in time and suffered irreversible consequences. Another illustration is spinal muscular atrophy, for which transformative therapies have become available within the last decade. Here you see siblings affected by SMA. The older brother was diagnosed at 6 months based on symptoms and began nusinersen treatment at that age. His younger sister, however, was diagnosed at birth because of her brother's history and began treatment at just 15 days old. The difference in their outcomes is striking, and it is explained solely by the age at which the treatment was initiated. 6 months for the older brother and 2 weeks for the sister. Is this treatment effective over time? Yes. Here are videos of the same siblings several months later. While the brother remains unable to walk, his sister is able to climb stairs, run, put her life in danger, riding bicycle. This demonstrates the importance of preventive treatment. Once irreversible damage has occurred, therapy can only halt further progression, not reverse existing harm. And here are the same siblings a few months later. Beyond SMA, many treatable conditions still escape current screening programs simply because no biochemical marker exists. Here is the case of a boy from Democratic Republic of Congo who arrived in Belgium at the age of 9 with severe hypotonia. After years of diagnostic odyssey, he was finally diagnosed with congenital myasthenic syndrome. Congenital myasthenia is a treatable disorder. Treatment is inexpensive, about €7 per month with salbutamol and pyridostigmine. And here you can see this boy, only after 2 days of therapy, he could walk independently. Had he been identified at birth, years of disability could have been avoided. Congenital myasthenia, therefore, was another example of a disease that could already be detected at birth today. However, there is a very wide range of treatable diseases that are still not included in newborn screening programs simply because no suitable biomarker exists. Nowadays, advances in sequencing technologies such as NGS, coupled with dramatic reduction in sequencing cost, now make it possible to consider genomic newborn screening. And this is how and why the BabyDetect project was born. The aim of BabyDetect trial was then to assess the acceptability and feasibility of implementing genomic newborn screening at birth in the general population. BabyDetect is conceived as a complement to conventional screening. In parallel with the sampling procedure, with a standard sampling procedure, a few drops of blood are collected onto a second filter paper, the gold card here on the right. In Belgium, the conventional filter paper enables the detection of 23 diseases, whereas the BabyDetect filter paper allows the identification of an additional 142 conditions. Both filter papers are centralized into a single newborn screening laboratory, where the two samples are registered within the same laboratory information system. This integration enables genotype-phenotype correlations for diseases covered by both approaches. The BabyDetect sequencing protocol is based on a target panel of 405 genes encompassing 165 treatable conditions. The 23 conditions included in conventional screening and the 142 additional disorders. These diseases span 10 different medical specialties. To be included in the panel, diseases or gene must meet mandatory criteria: a pediatric onset before the age of 5 years, well-defined pathogenic or likely pathogenic mutations with high penetrance, a severe natural course leading to significant disability if untreated, and the availability of an approved therapy or an ongoing clinical trial. The laboratory workflow of BabyDetect largely mirrors standard NGS procedures. DNA is extracted from 3 3mm bunches. Libraries are prepared using Twist Bioscience reagents. Sequencing is performed on an Illumina platform, and data are analyzed using an internally developed pipeline and ALISA Interpret software. The whole newborn screening workflow distinguishes between a screening component represented on the left side of the schematic and a diagnostic component on the right. In the BabyDetect screening arm, a negative sequencing result leads directly to a negative BabyDetect screening. In contrast, a positive sequencing result is systematically compared with the outcomes of conventional screening. If the condition is part of the conventional program and both results coincide, the BabyDetect screen is deemed positive. If the condition is not covered by conventional screening, the BabyDetect analysis is repeated in its entirety to exclude potential sample mixed-up and/or analytical error. When both sequencing runs corroborate the findings, the BabyDetect screen is considered positive, and in such cases, the newborn and parents are recalled by the maternity pediatricians for confirmatory diagnostic testing on an independent sample. Which abnormalities are reported by the BabyDetect screening program? We sequence the coding regions of 405 genes, representing approximately 1.6 megabase. For each newborn, between 5 and 10,000 variants are initially identified. A variant prioritization algorithm implemented on the ALISA Interpret platform has been developed to report only class 4 likely pathogenic and class 5 pathogenic variants. Only pathogenic and likely pathogenic variants with a consistent interpretation across public databases, ClinVar, Franklin, and VarSum, are reported in accordance with the inheritance patterns of the relevant conditions. Variants of uncertain significance, the VUSIs, are systematically excluded, and copy number variations, CNVs, are not yet assessed. Our approach also is highly conservative, aiming to minimize the reporting of variants with variable penetrance, expressivity, or pleiotropy. The BabyDetect study is a population-based study that began at a pilot maternity unit in the Liège region. This maternity unit, part of a public hospital, records approximately 2,500 births per year. The hospital serves an extremely diverse patient population in terms of ethnicity and socio-economic background. The first baby was screened on September 1, 2022, and the BabyDetect project was subsequently expanded to a second maternity unit in the region on September 1, 2024. The study concluded on May 31 in this year, and participation was free of charge for all parents. And by the end of the study, 7,515 parents had been approached and 6,824 consented to enroll their newborns, corresponding to an acceptability rate of 91%. Information was provided to parents during the prenatal period in gynecology consultation offices, as well as immediately after birth in the maternity unit. Consent was primarily obtained electronically. Of the 6,824 sequenced samples, 134 tested positive. All of these cases received appropriate follow-up and medical management. Among them, 118 were for conditions not covered by conventional newborn screening, including 94 cases of G6PD deficiencies. 3 cases were identified as false positives, 1 primary hyperoxaluria, 1 SCAD deficiency, and 1 case of Wilson disease. We also report 2 false negatives, 1 case of progressive familial liver disease and 1 MTHFR deficiency. What did we learn from this pilot study? Let us first consider the example of primary hyperoxaluria type 1, in which we initially identified 2 class 5 variants. The newborn was referred for confirmatory diagnostic testing, and an independent sample was collected from both the infant and the parents for a family segregation study. This analysis reveals that both variants were carried by the mother, while the father carried neither. Additional investigation, including urinary oxalate measurement and renal ultrasound, shows no abnormalities. These findings confirm that both the baby and the mother are carriers of type 1 hyperoxaluria, but are not affected by the disease. This example highlights the critical importance of parental testing in confirming diagnosis identified through genomic newborn screening. Regarding false negatives, we reported 2 cases during this study. Both diagnoses were made following symptoms onset in these patients and in the context of workup by whole exome sequencing. We compared then the 2 variants identified by whole exome sequencing with the BabyDetect sequencing data. Both variants were indeed present in the raw sequencing data of BabyDetect, but were not reported because they had never been documented in any of the public databases before. Beware that genomic newborn screening does not rely on phenotypic information, since neonates are theoretically a healthy population. Consistent then with our conservative approach, we do not speculate on the pathogenicity of previously unknown variants, even homozygous nonsense variants like for TGP2. These variants have now been added to our internal databases for prioritization in future analysis. With respect to dominant inherited diseases, several cases of hypertrophic cardiomyopathy were also identified. We reported 5 heterozygous MYH7 mutations and 2 heterozygous MYBPC3 mutations. In one reported case, the father was subsequently found to have previously undiagnosed signs of cardiac hypertrophy. This raised the question among specialists as to whether late-onset forms should be reported. Indeed, with the exception of de novo mutation, dominantly inherited diseases should, in principle, already be known within affected families, since one of the parents is at least theoretically affected. After consultation with pediatric cardiologists and geneticists, it was decided that such dominant forms would no longer be reported and that only recessive conditions would be disclosed. Our variant classification framework was accordingly revised. The case of CPT2 deficiency illustrates the complementarity between conventional and genomic newborn screening. In one infant, we identified two pathogenic variants in the CPT2 gene, while conventional newborn screening results were normal. Orthogonal confirmation analysis on an independent sample, including CPT2 activity in cultured fibroblast and plasma acylcarnitine profiling, confirms the diagnosis of CPT2 deficiency. The availability of the BabyDetect results allowed for accelerated clinical management when the infant developed symptoms associated with the myopathic form of CPT2. It has been demonstrated that screening based on biochemical markers for inborn loss of metabolism has greater sensitivity and specificity than genomic screening. Moreover, the turnaround time of genomic screening is currently not compatible with the clinical onset of metabolic diseases. It is therefore evident now that at present, genomic newborn screening should be regarded as a complement to conventional screening. It is not aimed to replace it. The turnaround time consistently improved over the course of our study, decreasing from around 60 days at the beginning to 22 days by the end. This progress was made possible, among other things, by the inclusion of a second maternity unit in September 2024, which significantly increased our throughput. We also compared our results with other pilot studies, notably the Guardian study in New York City, which sequences a smaller number of genes but reports a higher rate of positive cases, 3.7% compared to 1.9% in BabyDetect study. The number of false positives in BabyDetect study was lower than in Guardian study, reflecting BabyDetect's more conservative approach, whereas the Guardian study also reported those. Accordingly, the positive predictive values of BabyDetect was 98% compared to the 83% of Guardian study. The BabyDetect and Guardian studies are not the only genomic newborn screening initiatives. Approximately 30 similar projects are currently underway worldwide. Moreover, some regions have already integrated genomic newborn screening into their official newborn screening programs. For example, the Puglia region in South Italy has already sequenced around 10,000 newborns since March 2025, while United Kingdom, Florida, and Abu Dhabi have recently announced their intention to formalize such program in the near future. What are the achievements and future perspectives of the BabyDetect project at the end of the pilot study? The BabyDetect test has recently been accredited under ISO 15189, and it is also registered as a medical device by the competent authority in Belgium. We are planning to automate the process and are actively participating in the International ICONS Consortium to analyze gene lists. A transition towards whole exome sequencing is planned, along with the development of a second-tier confirmatory methods to obviate the need for family segregation studies. In addition, the availability of the BabyDetect test has been extended to other maternity units in our region. In conclusion, genomic newborn screening is feasible and accepted by population, but it raises also a wide range of questions and faces numerous challenges. These include economic considerations, who will bear the costs, as well as clinical and societal issues. Newborn screening is undergoing a paradigm shift. Should it remain a public health program or evolve into an individual preventive medicine test, such as for cardiomyopathies I described previously? Will it ultimately lead to a genetic passport for the entire population? Major challenges also concern data protection and storage, including the risk of genetic discrimination, the unexpected discovery of familial relationships, and the long-term preservation of massive datasets. Psychosocial risks must not be overlooked either. particularly parental stress in response to false positive or ambiguous results, as well as the potential of stigmatizations of certain groups within the population. I will conclude here by expressing my gratitude to all members of the BabyDetect team, as well as to the sponsor who made this project possible. For further information, you may consult our publication reporting the interim results or contact me directly by email.
Thank you so much. It has been really, really interesting. And we have some questions. Actually, we have a lot of questions.
Okay.
So how are the initial gene condition pairs are created?
How were the genes? So
The gene condition connect, yeah.
Yeah, so we had the discussions, meetings with several pediatrician specialists in the hospital where was a pilot maternity, and it was discussed to consider which genes or disorders were already treatable, and we decided to include the genes in our list. So over the 3-year project, we had several meetings with clinicians, and the first version of our list has evolved to a version 2 of the list, where some genes were removed because it was not very clear how to manage these patients, and other new genes were also included because it was clear that a treatment could be available. So, for example, the retinoblastoma gene was added in the version 2 of our panel. I did not explain all the version of the gene list because it was too long to explain. But yeah, the gene list is it's a long debate, and in the different initiatives worldwide, so I explained there are about 30 initiatives, only in total more than 2,000 genes are included in the different initiatives, and only 20% of genes are included in all initiatives. So there's a wide variety of genes, and that's the aim of ICON's consortium to try to harmonize a consensual gene list for genomic newborn screening programs in the future.
Perfect. There is 2 other questions around this topic. So one is how to address novel variations when you see them on the newborn screening, and also when you have new ones, if I understand the question, when you have new ones, if this is submitted in ClinVar.
Yes, we try to submit them in ClinVar, but only when we have, as I mentioned, we do not report variants that have never been described previously in the literature. We only report very well classified variants because we do not want to generate stress or harm, because that would kill the project if we could report VUSIs for all babies, because we identify VUSIs in all patients, but we do not want to harm or to stress the whole population. So we want to be very conservative. And in the 2 cases I presented with false negative results, yes, they have been finally described as pathogenic or likely pathogenic variants, so they are now added to public databases. Yes.
Okay, perfect. Also, we have people coming from the US. So one person is asking if the follow-up testing is also covered on this study. He understands that the coverage is different from the US and in Belgium, but I don't know if you can have an insight around that.
Yeah. So all babies identified by the BabyDetect study have been referred to pediatrician, and a follow-up has been initiated to confirm the screening results by diagnostic test with orthogonal methods, or by NGS also. It depends what kind of assay or test would be available. And in case of confirmation, so all babies were treated or managed or under surveillance based on the considered disorder. But this follow-up is not our sponsor were sponsoring the study only for the screening part of BabyDetect, and then for the diagnostic part, it is covered by our national health insurance. So all patients benefited from this reimbursement of the test of the confirmatory test. So.
Yeah, actually, this was another question that someone just put. If you envision if this could become part of the Belgium national newborn screening program in the future, but if I understand, you just answered that. Like if it would be part of the biggest like Belgium.
Yeah, we have already approached our political and authorities trying to asking for a future reimbursement or coverage by health insurance, national health insurance, but that will take time, a very long time, because that's, as I mentioned, it's still very expensive. So that will take years. So yeah, but it's an ongoing process. We already also have approached the private health insurance, but it's an ongoing process.
Yeah.
Okay.
Good. We are gonna do one more question, and then there is a couple of others that we will answer by mail if that's fine. So it mentions that involving parents who confirm the positive screening results, it could be challenging, we assume. So do you think that there is other strategies that can help you to streamline the process of confirming positive screening results, or actually even eliminating the need of this direct parent involvement?
Yeah, that's one of our next objective is to implement long-read sequencing as a second-tier test, so to avoid the implication of parents. So if we have a positive screening results before reporting the result, we would like to implement long-read sequencing second-tier test on the first filter paper we received to check or to assess the phase of the two variants.
Perfect. Okay, so we will leave it here. Thank you so much.